Hey guys, we're going to be starting topic 3.4 and 10.2, which is theoretical genetics and inheritance. We're going to be discussing all the different, or many different types of inheritance and how that occurs between parents and how it's passed on to offspring in predictable ways. So just as an introduction, a man named Gregor Mendel, who was a monk, he was kind of the father of the understanding of inheritance. He discovered patterns of inheritance before even the discovery of chromosomes or genes. And he did this by comparing inheritance patterns of pea plants. And he looked carefully at 7 different characteristics and was able to find that things are inherited in predictable and mathematic ways. So he is, kind of who we're going to be modeling our study after. We'll look at how he did some of his methods and how they contribute to modern inheritance knowledge. So kind of a lucky mistake for him because he unknowingly chose genes that are controlled, characteristics that are controlled by single genes in the pea plant. And so it made his job a little bit easier, a little bit less confusing when he was determining how inheritance worked. So before we dive in, I want you to take a moment, read understandings 3.4.1 to 3.4.4 and 10.2.1 to 10.2.4. After you've taken a second to read those, I also want you to think about how many alleles of each gene does the offspring receive, and where do the different alleles come from? And also remind yourself, what is an allele? Okay, before we get real deep into the inheritance patterns, we need to have our vocabulary set. So here is a giant list of several terms that are going to be used over and over and over again throughout this topic, and you need to be sure that you are familiar with these. Feel free to take notes on them, make flashcards, etc., because like I said, these terms are terms that are going to come up over and over again, and you need to know what they mean immediately. In addition, they'll continue to show up through the remainder of this genetics topic. So the first is genotype. Genotype is the alleles or the genes that an individual possesses. Phenotype are the physical characteristics of that organism. So for example, someone may have a gene for a widow's peak, but they may not be expressing that widow's peak. So in that way, genotype and phenotype can differ. However, genotype, of course, leads to phenotype. We've also got 2 categories of alleles, 3 categories, I guess, but we've got dominant alleles, which are those that will always be expressed, whether they're there from mother and father or just from one parent. And then recessive alleles, these are those that are expressed only if inherited from both parents. Therefore, if a dominant allele is present, the recessive allele will not be expressed. And then there's also codominant alleles. These are alleles that will both have an equal effect on phenotype and sometimes actually result in a mixing effect. For example, maybe we have a gene for a red flower from one parent, a gene for a white flower from another, and the offspring ends up being pink. Locus is the particular location of a gene on a chromosome. This is important for being able to match up homologous pairs and also to make sure that the offspring gets a complete set of DNA. Homozygous, homozygous inheritance is a situation where the offspring has inherited identical alleles from both parents. So, for example, maybe we got a recessive allele from mom and a recessive allele from dad, then we'd be homozygous. Heterozygous is when different alleles are inherited from each parent. So perhaps you get a recessive allele from mom and a dominant allele from dad. From there, a carrier might be an individual that possesses a recessive allele for some trait, but it is not expressed in their phenotype, and so could be passed on. So by definition, this person would have to be heterozygous, being a carrier but not an expressor. And then lastly, a test cross is something that we'll do a lot of. This is a sample crossing to test a suspected heterozygote by crossing it with a known homozygous recessive individual. Because then if that individual is heterozygous, then the dominant trait will at some point arise. Okay, so keep in mind, alleles are the different variations of a gene that can occur. For example, if we talk about the pea plant example, peas can have either green peas or yellow peas. So those would both be a gene for pea color, but different alleles. So, alleles come from the different parents. There's 2 alleles for each gene, and they separate into different gametes during meiosis. So remember, during meiosis, we do have a reduction division in meiosis 1 that separates these homologous chromosomes. And so now there's just one allele here, one allele here, and then of course sister chromosomes again get separated. But this happens during the production of gametes, which are sex cells, and they are the inheritable cells from the parents. So gametes are haploid, containing only 1 allele for each gene they contribute, rather than 2. What this allows is for when 2 haploid gametes fuse together, so if they each have half of the DNA, so we'll say that's N plus N, then the offspring is 2N. Ooh. 2N. There we go. So gamete production is very important, and also keeping track of how alleles move from parent to offspring. A couple of things to keep in mind when we talk about fertilization is that fertilization is a random process, with each allele from one parent having an equal chance of fertilizing any other gamete of the other parent. So remember how we talked about last topic, how there are thousands and thousands and thousands of variations of allele combinations that offspring can receive from one parent due to random assortment of homologous chromosomes and crossing over. So the egg that's selected, or the sperm, or the egg in the pollen grain, depending on what species you're discussing, there's an equal chance for any of those alleles to be fertilized by any other allele. So again, variation in offspring is enormous, and that's why we have so many unique individuals. One allele for the offspring comes from each parent, and one allele will be dominant over the alternative factor. So in most cases, other than codominant situations, one of the alleles will be dominant and will overshadow the other factor. When we talk about inheritance, it's important also to know the location of the allele or the gene. Genes that are carried on a particular chromosome are called linkage groups, meaning that they will likely be inherited together. However, chromosomes of a linkage group can be traded through crossing over. So linkage groups will likely be inherited together, but there definitely is some uncertainty with that depending on the amount of crossing over or where it occurs. So that's one thing that we're going to look at that causes some confusion when we talk about inheritance. However, unlinked genes are much easier to follow as they segregate independently during meiosis and are therefore inherited on their own or separately. So unlinked genes make things a little bit simpler when we talk about predictable inheritance, but we will talk about linkage groups as well. Okay, so like I said before, Gregor Mendel, who was a monk, was kind of the father of inheritance. So he discovered the principles of inheritance by carefully observing the crosses between pea plants. He began his work in 1856, and he studied the inheritance of 7 characteristics that were unlinked, actually. And so he was able to use purebred plants and do test crosses to predict the way that a monohybrid cross would work, which is a cross where you're just considering one gene or one set of alleles. I have a short video that I'll show you about him in class, but these are the 7 traits that he focused on, and they resulted in some really incredible information that has largely influenced our understanding of inheritance to this day. So like I said, all of Mendel's observed traits were controlled by just one gene, so there was discrete variation, either wrinkled or smooth, either yellow or green, etc. However, a lot of the traits that we'll discuss, especially when we talk about like human physical appearance, etc., are polygenic traits, meaning that they're controlled by multiple genes, and so instead of having one or the other, they have a continuous outcome of observed phenotypes. So for example, shade of skin, there's every variation that you can imagine as far as being lighter or darker color in skin, and this is due to polygenic traits. So, something to consider. This is just, like I said, a brief introduction to our inheritance topic. The remainder of the topic will be spent completing Punnett squares and pedigrees for different types of inheritance. Please be sure to read pages 110 to 123 and 313 to 326, and have a great day.